Today we're going to be talking about the nervous system. The first part of the nervous system is called the central nervous system, or the CNS. Here I'm drawing a little person, and in green I'm going to draw the CNS, and this is comprised of the brain and the spinal cord. This is sort of the central processing center of our nervous system. Information comes in and out of the CNS via the peripheral nervous system. These are all the nerves that extend from the CNS out to the periphery, so out to the muscles, and back from the periphery or sensory structures like touch sensors in our fingers back into the CNS. Here I'm drawing it, the PNS, the peripheral nervous system, in red. If we zoom in on the nervous system, we can look at the building blocks that comprise this organ system. And like other organ systems, we find biological cells. You're probably familiar with this drawing of a cell with a cell body and a bunch of organelles. And a neuron is just a specialized cell. It has many of the same organelles, and it also has a cell body. But off of the cell body, we have specialized structures. In this case, these blue branching structures are dendrites, and they receive inputs into the neuron. Information gets passed into the neuron via the dendrites. And then we have an axon, which is where information is output, in purple here. Another name for the cell body of the neuron is the soma. So with the neuron, we have information that gets passed from left to right, as I've drawn here, from the dendrites through the cell body out to the axon. This information is then conveyed to downstream neurons and other cells, like muscle cells, at a junction that we call a synapse. So here we have two neurons. The neuron before the synapse is the presynaptic neuron, and then the postsynaptic neuron is after the synapse. In this case, we're going to be focusing on chemical synapses, but there are other types. And there's no physical contact between the presynaptic and postsynaptic neuron. But in the presynaptic neuron, we have these vesicles that are filled with these orange neurotransmitters. When a burst of electrical activity reaches the end of the axon here, the synaptic terminal, the vesicles are released at the surface, releasing neurotransmitters into this gap here, the synaptic cleft between the two neurons. The neurotransmitters travel across the gap to reach the postsynaptic neuron in blue and bind with receptors. These blue receptors then, when bound to by the neurotransmitter, cause a change in the postsynaptic cell so that the information can be passed along. Generally, we talk about this information being passed in the form of electrical activity. And you might be wondering to yourself, how does the cell generate and transfer electrical activity? So let's zoom in on the cell membrane and look at what's going on at rest. We're zooming in on the dendrite, but this is really going on anywhere in the neuron. Here's our cell membrane, a lipid bilayer. We're going to look at the resting conditions both inside and outside of the cell. Outside of the cell, we see a high concentration of sodium ions. Here I'm drawing them in red. There's some sodium inside the cell, but it's highly concentrated outside. In green, I'm drawing the potassium ions, which have a much higher concentration inside of the cell. We also have a high concentration of negatively charged proteins inside of the cell, which I'm drawing here as yellow A's. So this leads to a charge difference across the cell membrane. So we have a more positively charged environment outside of the cell relative to the more negatively charged environment inside of the cell. So this charge difference across the membrane is called the membrane potential. And at rest, we call this more specifically the resting potential. And we talk about potential because the built-up difference in charge is an unstable state. Biochemically, things want to move towards a stable, uncharged state. So the membrane is keeping this system in an unstable state. So why do we have this imbalance? So the main culprit is the selective permeability of the membrane. Potassium ions can pass relatively easily through the membrane, but sodium ions have a much lower permeability. And our proteins, these negatively charged proteins, cannot pass through the membrane at all. So there are lots of different electrochemical forces at play. Ions wanting to move down their concentration gradients, and ions wanting to move down their electrical gradients. And when these forces balance out, we get this difference in voltage that we see at rest between the inside and outside of the cell. We also have a transmembrane protein, the sodium-potassium pump, that also contributes, although at a very local level, to the membrane potential. This, using energy in the form of ATP, moves 3 sodium ions outside of the cell for every 2 potassium ions it moves into the cell. So this contributes to this imbalance. So now we have this built-up potential energy. Sodium wants to flow down its concentration gradient because of diffusion into the cell, and potassium wants to flow out of the cell. But due to the electrical gradient that's built up, we stay at this resting potential. This is all thanks to the semi-permeability of the membrane. So when a stimulus disrupts the cell at rest, things change drastically. We'll talk about the stimulus being from another neuron at a synapse, but it could be due to an external stimulus on a sensory neuron. For example, the neurons embedded in your skin respond to being deformed. That's how we can sense touch. And that deformation causes an electrical change in that cell. A stimulus at the synapse can lead to an opening of special sodium channels that do let sodium flow freely down its electrochemical gradient across the membrane. When this happens, we see a huge positive spike in the membrane potential as the positively charged sodium ions flow into the cell. So now I'll draw how the membrane potential changes over time as these processes occur. So we start with a negative membrane potential at rest. The inside of the cell is more negatively charged than the outside. As the sodium channels open, we increase our membrane potential, and when we cross this dashed line, which is our threshold for excitation, then the process continues and can't be stopped. So this is an all or none event. Once we cross the threshold, it occurs. And so the membrane potential increases, and at this peak here, the sodium channels close, and a different type of channel that allows potassium ions to pass freely opens. So these potassium channels open, and our potassium then can rush outside of the cell. This returns the cell to a negative membrane potential. We overshoot the resting potential a little, but quickly we return to that initial resting potential. This whole process is called an action potential. This is a spike of electrical activity that can traverse down the neuron. Each phase of the action potential has a name. Here I'm writing those out. The initial increase in our membrane potential is called the depolarization, because we're going from a polarized state at -70 here up towards 0 and into positive. Then we have a repolarization where the cell becomes polarized again. That little overshoot at the bottom is called hyperpolarization. So then we get back to the resting state of our cell. That resting potential reestablishes itself because of the baseline selective permeabilities of the membrane. So this action potential can get passed along the cell membrane to traverse the length of the neuron. We see these all or none action potentials most prominently in the axons. They sort of build in the dendrites and cell body. And so we can look at these spikes of electrical activity, and we see this is how information can be coded and passed within the nervous system. Just a few words about these neurotransmitters that convert the electrical signal from the presynaptic neuron into a chemical message that then can be converted to an electrical signal at the postsynaptic neuron. These neurotransmitters in the synaptic terminal are a limited resource. So when they're released, we can see one of 3 things happen. They either drift away, they can get broken down by enzymes, or they're reabsorbed via a process called reuptake. And here I'm drawing a little reuptake transmembrane protein that will pump these neurotransmitters back into the presynaptic cell. These neurotransmitters can have different effects. Some are excitatory, transferring a signal like I've described, but some are inhibitory, and they reduce this downstream signal or the sensitivity to signals from other neurons. These neurotransmitters also have global effects. So if we bathe a bunch of synapses with different neurotransmitters, we can mimic the effect of activating those synapses. For example, if we give someone dopamine, it can activate synapses that utilize dopamine as a neurotransmitter. This could result in an increase in pleasure and a depressed appetite. The basis for many psychiatric drugs is affecting, mimicking, or interfering with neurotransmitter action. There are 2 specific classes of drugs that we'll talk about. And so if we look at how these act on the receptors, I'm going to blow up these receptors. We can have neurotransmitters that act as agonists, and these are chemicals that mimic a neurotransmitter at the receptor site. So in this case, it locks into the receptor, activating it. This will mimic the action of the neurotransmitter, strengthening its effect. The other drug class are antagonists, and these block the action of a neurotransmitter, here physically blocking the receptor from access by a neurotransmitter. In class, we're going to talk about some more details about neurons and the nervous system, including myelin, which is a substance that coats some neurons, and the parasympathetic versus the sympathetic nervous system.